introduction, and welcome everyone. Thanks for coming out first thing in the morning to listen and learn about science. Really appreciate that. Like Adam said, I'm the CEO now of Treehouse Biotech, and today my talk is going to be on the lock and key, understanding the molecular mechanism of action of cannabinoids. And so I want this to be a scientific discussion, but also an introduction to a broad audience into what this model is, what the cannabinoids, how they function in this model, and then what the endocannabinoid system is. So I'm going to start off with the lock and key model, which actually dates back more than 100 years and is the basis of modern medicine, really. So talking about active site binding and ligands and receptors, and the metaphor is going to be throughout this talk, locks and keys. And so kind of off the bat, you have a lock and keys, go into it and unlock it. And so get that in your mind. And then we're going to dive into kind of linking that to the actual science there. I'm going to go into the cannabinoids then, talking about what's the difference between endocannabinoids, phytocannabinoids, and when you hear CBG, CBGV, CBD, delta 8, THC, what all those acronyms really mean from a chemical standpoint. And then diving into the endocannabinoid system, what we know so far, kind of what's unknown, and talking a bit about the entourage effect, which hopefully kind of brings the whole talk together. And so, like I said, I want this to be accessible to a broad audience, so hopefully I can accomplish that. And then just a brief background on what we do at Treehouse and why I have some knowledge of this. So we founded back in 2015. We're out of Colorado. And from the R&D side, our biotech team is very focused on the isolation of minor cannabinoids, which we'll be talking about. Converting different cannabinoids between one another chemically, as well as then studying individual cannabinoids and what they do as medicine. And then the other side of our company is a kind of wholesale production facility that does different isolates, extracts, and things like that. So getting into the history now of this lock and key model, 1894, Fisher was a very famous chemist, discovered a lot of things about all the sugars we have today, and was doing this at a time before we really knew much about chemistry and bonding and structure. And so the proposal was that molecules that are, every molecule out there has a different three-dimensional structure, and that is what gives them their properties. And within the body and within all of biology, those structures fitting into specific locks, we'll say, those keys fitting into locks, is how chemistry and biochemistry happen. So that was the kind of historical view, and obviously science has advanced over 100 years now, and there's a lot more known about it, but generally speaking, this is still the model with a little bit of added details. And so it's a highly specific model, so every little change in this three-dimensional structure here, every little direction of those bonds, what atoms are there, actually affect the whole kind of biological outcome of that molecule going into a system. So these are the keys, the individual molecules, the drugs, the food, anything you're putting in your body is acting like some sort of key. And then the locks are all of these proteins, which are commonly shown like something like that. And there's hundreds of thousands of individual proteins all working together at any given time that makes life possible. So that is biochemistry. And this is the basis for modern medicine and drug development. And so all of the pharmaceuticals, all of the nutraceuticals, all of the food you eat, this is the model for how those individual molecules interact. And really, if you find your specific lock within the body that is responsible for a disease state, modern pharmaceuticals is finding the right then key that interacts with that lock to either unlock it, keep it locked, whatever the metaphor is there, and you can specifically then treat disease states with one specific molecule. So that's the background, the history of the model, and then some terminology, because I've probably already thrown around a bunch of words. The lock, when we're talking about, is a protein or receptor. Those are the kind of biological terms. The key is a molecule, or you'll hear ligand, and if you're kind of wanting to go learn more about this, ligand for the protein is an also interchangeable word. And then we have the endocannabinoid system, which is the system within your body, which is comprised of a lot of those keys, those receptors throughout the body. And that system is your endocannabinoid system interacting in the body in all mammals. And then the different cannabinoids, we'll talk about that later on. But A cannabinoid is either a phytocannabinoid or an endocannabinoid, and we kind of shorten it because you don't have to add all the extra syllables. Endocannabinoids occur naturally within you. You're making it right now. It is responsible for your current endocannabinoid system. Phytocannabinoids come from plants, and so that's the difference there. So getting back to this model now, you have, say, 2 keys and 2 receptors, and kind of highlighting this key is different than that key, and we've all, you have a whole keychain and you're trying to unlock your door, and one fits perfectly, and a lot of them kind of get in there and don't work. And that's the true kind of nature of this, but chemically what we're talking about is a very different actual lock, right? So you maybe have seen something like this, a picture of a cell in your life. Maybe it was biology in high school, maybe in college, or you're just not very familiar with it. But a cell you can see with a microscope. It's very blurry. It looks like a blob at that point. But anything kind of higher resolution than that is very hard to actually see with a microscope. And so now we're zooming in from this picture over here to this picture right here. So this little orange layer on the entire outside of this cell is now zoomed in here. So this is the bilayer that separates the cell from the outside and the inside. And within this bilayer are all these little globs, which is shorthand for these keys, or these receptors, these locks. So this is what's happening when you're taking a drug. It comes in, floats in, interacts with this receptor lock, sorry, gonna go back and forth. The lock, and then unlocks it and stimulates some sort of chemical reaction, and that's how biochemistry happens. And then zooming in a bit more, all of these different receptors have a different structure, and they're very complex three-dimensional things. And like I said, there's hundreds of thousands of individual proteins, receptors within your body that keep you moving, alive, breathing, thinking at any given time. So that's Biochemistry 101, how we get from the cell and very much zoom in. And now to kind of give a dynamic view of this, this is going to be a video of one specific receptor, one specific lock, and then I'll highlight molecularly what you're looking at. So this is now a three-dimensional representation of one of those little locks sitting in that membrane, and it's going to zoom in here into a bunch of different molecules being inserted into the key slot. Give it a second. So this is now your key slot right here within the protein, and it's showing just random molecules being fit into that and showing you how they fit three-dimensionally. And so this is how drug development works. They'll make 100,000 different molecules and screen them against this specific receptor and see which one's a hit right here. So that fits really well in there. So now you can analyze how this fit is working. And this is kind of, again, just a dynamic view of what's really going on from a molecular standpoint. And you don't have to understand the carbons, the hydrogens, all of the bonds and stuff. But you can see, I think, three-dimensionally and structurally that specific things will fit differently into these receptor sites. And so that's kind of the basis of this lock and key model and how we're going to start now talking about cannabinoids in particular. So everyone's probably heard of THC and CBD here. We're at the CBD Expo, and I'm going to now highlight the only difference between these two molecules. So to somebody who's not very trained in chemistry, it might even be hard to pick out the difference between picture 1 of THC and picture 2 of CBD. It's right here. It's this little orange thing attached to this thing. It's one extra bond. So in CBD, that bond is not there. That is the only difference between CBD and THC, and that is responsible for all of the downstream effects. So that's how powerful this molecule is, right? Or this model is, that three-dimensionally, that little change is so impactful. And so how you can kind of visualize it is, in CBD, you have a bond between these two halves of the molecule, but they're free to rotate about that kind of axis, and so you have this really kind of floppy, jiggly molecule. With THC, you have one extra bond. That's the one I pointed out. So you now locked an extra bond, and you can no longer rotate. You're stuck in this flat geometry. And that's it. That's the whole change in how they affect the CB1 receptor and the entire, you take CBD, don't get high. You take THC, you get high. So it's pretty profound, and to me always impressive, even today, that such a small change can have such a broad impact on your actual perception of the universe, frankly. THC puts you in a different mind state, and it's just one bond. So I guess I should have done that first, but that's kind of highlighting now in the model how the keys are changing. Now to scale representation of this huge thing right here is the receptor. It is the CB1 receptor. That is a crystal structure of the receptor. And to scale, that's roughly about how big THC or CBD is. So these tiny little things are going in and then causing all of the effects downstream, whether it's medicinal or recreational or anything like that. So it's a pretty profound change for such a small little three-dimensional structure. And then to also consider, you can just put, you can take your cornflakes and your milk and your whatever you're gonna eat in a day and all the vitamins, and none of those molecules, hundreds of thousands of different individual molecules, do exactly that. So it's that one molecule that has that very specific effect. So biology is remarkably selective, and it's very impressive. And then I'd also like to highlight here that this is a 2016 paper in the journal Cell. So that's very recent in scientific terms. And this was when the CB1, that receptor, we knew the structure of it. To this date, we still don't have a crystal structure of the CB2 receptor at all. So even scientifically, we're at a very cutting edge point where there's a lot of things to be learned from the basic science level, aside from just the medicinal effects of CBD and the other cannabinoids. So now moving forward, I'm going to focus on the keys. This is going to be the key portion of the talk. We talked about the locks. So what is the difference between phytocannabinoids, endocannabinoids, and how can we start to understand those differences? I don't know. That's not supposed to. Oh, that was supposed to be a picture, so it didn't translate well into this presentation, but that's okay. So we don't have pictures of the endocannabinoids. So we're going to go right on to the phytocannabinoids. So the molecules actually look very distinct, the endocannabinoids and the phytocannabinoids, and I apologize, I can't show you that right now. But now I'll just focus on why the phytocannabinoids, you can even, again, as an untrained chemist, CBD, THC, maybe you can now see a little bit clearer. This is a two-dimensional representation versus those 3D ones, because those get very cumbersome. But that extra bond right here, that's the only difference there. CBN looks very similar. You see a bit more of these double lines, which is kind of, we don't have to get into the chemistry there, but it's just a different bonding structure. But CBG and CBC, they look pretty distinctly different. And so you could expect that the medicinal effects of those will be pretty distinctly different as well when compared to THC and CBD, because the structures are so different. So moving forward now, I'm going to go into what I call a CBD's case study. And so depending on your background in chemistry and your knowledge of this industry, people will be, I want more CBDs in my product. I want different CBDs. How many CBDs do you have? And so trying to get away from that true misnomer, there is only one CBD. This molecule, that specific structure, that key is CBD. There are what are called isomers, or very similar molecules that share the overall kind of the CBD portion of it. And I'm going to highlight the little changes that affect the naming so that everyone here can go into a conversation and there is not multiple CBDs. There are different types of very similar molecules, but they all have different names and different structures. So this is CBD, and I'm highlighting this chain right here, and that's just a couple atoms connected together. That's 5 carbons connected. CBDV, THCV, CBGV, the V stands for varin, and all that's saying is that there's only 3 carbons on this little tail. So whenever you're hearing the V's, that's really what it's referencing from a structural standpoint. And as I just showed, one bond can make a pretty profound difference. So CBD and CBDV could have very different effects. They could have similar effects, and frankly, it's very hard to predict what those effects will be because of the complexity of the biochemical system. You pretty much have to study them independently to really tease out the differences between those 2 molecules. Next, there's CBDA, there's, again, THCA, CBGA, and what the A is referring to in those is the acid. And so highlighting here, there's nothing there. There's 1 hydrogen there, but that's okay. Here, there is a carboxylic acid group. Again, don't have to know chemistry, but that's what the A stands for. It stands for acid, and acids are very common throughout all of biology. They help kind of the water solubility of all of life. So there's DNA is a nucleic acid. There's fatty acids, which make up the membranes. There's vinegar, acetic acid, and so the word acid, I think, in general has a kind of negative connotation, but if you think about it biochemically, it's a very common thing. So that's the only difference between CBD and CBDA. And you can convert one to the other, so this is kind of now your cannabinoid conversion with heat. So if you heat CBDA at, say, 120 degrees Celsius, this acid part just pops off, turns into carbon dioxide, floats away into the atmosphere, one more greenhouse gas for planet Earth. But you can also just over time sitting there, it will happen slowly. And so that's the only difference, and that's kind of actually nature. The plant synthesizes all of the cannabinoids as acids first, but by the time they are usually consumed or are active in humans, they have lost that acid portion. Now getting into, you may have heard of delta 8 THC, delta 8 CBD, delta 9 THC, which is the main THC component in marijuana that gets you high and is the kind of class 1 DEA controlled substance, is delta 9 THC. So the delta is referring to, the delta number is referring to where this little double bond is within, say, a ring structure here. So the double bond moves from here to here, and that's the only difference in the naming. But again, it's not the same molecule. It is not that CBD. It's now a new molecule with potentially very different medicinal effects. And so they seem like very small changes, but the first thing I highlighted was the small change between THC and CBD is one extra bond. So any of these changes here could be very profound. And now when you multiply this, so this is 4 different isomers of, say, CBD, but you have those same 4 for CBG, those same 4 for THC, same couple for CBC, same for CBN. So now you're talking about a field of 25 different cannabinoids. And when you're looking at, say, an extract, you have all of those interacting together. So it's very hard to piece out which portion of that puzzle is really responsible for the medicinal effect. So it gets very complicated, but hopefully now we can all discuss and you can go walk around the trade show floor and have a better idea of what people are talking about when they talk about these cannabinoids. So I mentioned maybe 4, 8 cannabinoids there. There's 100 naturally occurring cannabinoids that are known. And a lot of them are these very small changes. And so here, you're adding some oxygens, and this structure looks pretty different. I mean, these two look pretty similar. One has an extra oxygen, one doesn't. This one has a shorter side tail, all of these little random structural changes. But any one of those 100 naturally occurring cannabinoids could have a profoundly different medicinal effect. And really, maybe one is great for anxiety, one is great for sleep, or maybe none of them truly are. And we don't know that, and the research in this field is in such an infancy because of laws in the past 30, 50 years that not a lot of that is known, frankly. And so a lot of times people will ask me, what does X cannabinoid do? And my best answer is, we don't know. And so CBG, CBC, there's a handful of studies. I'm talking like 10, 15 maybe. On all of these ones I put up here, there's probably 0 to maybe 1 study on any given one of these molecules. So from a true scientific standpoint, there's so much to learn, and very much all of the other stuff you hear, I would say, is a lot of anecdotal stuff that's being propagated through the internet that doesn't have scientific backing. And so I'm very adamant about not making false claims in that sense, but also advising people to don't believe something if you read it on the internet. That should be a general rule in today's day and age. But also, until there's scientific studies that are reproducibly proving some sort of claim, it's not, scientific. It could work, and not also say that, if somebody takes a hemp extract and their pain goes away, I'm not going to say don't take the hemp extract. If it's working for you as a nutraceutical, great, but it's not. We don't understand why it's working. So that's a pretty big difference, right? So now going into, okay, again, we lost an image here, but that's okay. So we're going into the endocannabinoid system, and this text is going to be impossible to read, so I won't worry about it. But the CB1 receptor, so now back on the lock side of things, is localized primarily in your central nervous system. So it's in your brain, your spinal cord, and is really responsible for a lot more things like memory, your motor control, your appetite, your hunger, things like that. So it's a very brain-focused receptor. The CB2 receptor now is localized kind of everywhere else in the body. It's also in the brain, but it's more focused on inflammation and immune response. So CB2 is very, immune response focused, and that's why it's all throughout your body. And that's, in your brain, it's specifically for immune response. And then inflammation, which is a very broad topic because anxiety, stress, actual physical inflammation, pain, is all from the same system. It is inflammation, whether it's a kind of mental state of inflammation or not. So that's what the CB2 receptor is for: inflammation, immune, CB1, memory, cognitive, things like that. And so looking at, actually I'm going to go back a bit. So just to kind of recap, THC works on CB1 and CB2, which is why you get the mental effects, the high of it. And CBD only works with the CB2 receptor, which is why you don't get high. And again, just that one bond difference. And I'll go into that in a bit. But to look into what is going on, so now we're looking at this membrane again with this receptor here. And I'm not going to explain all of this, and you'd have to have quite a bit of background in biochemistry to even have a chance at understanding this. But I will walk through it so you can appreciate the complexity of biochemistry and medicine in general at a molecular level. So this kind of yellow thing right here is representing your key coming in and hitting the lock receptor. So once that happens, you get all of this in the cell happening. And so, I mean, these little blocks and squares and things are all different proteins doing different things. So each colored block is a completely different protein that has a different function. So as soon as this happens, say it turns this off and that on, which is why this is now turning this thing on and off, and that's turning that on, and very complex, very quickly, and all of these acronyms are different molecular processes that have been studied independently. And so you're getting this turning into that, which makes this go to, it's massively complicated. And so for every individual molecule hitting the receptor, there's a different downstream effect, which is why biochemistry and drug development is so complicated, because one molecule doing this here could have a completely effect somewhere else in your body. And really, I think when you see a pharmaceutical commercial, all of those side effects, like that's what you're looking at here, is the true complexity of biochemistry and how your body self-regulates itself. So now we're going to look at a kind of case study of the CBD THC, which I think I highlighted a bit previously, but I'll kind of hammer it home again. So comparing these two molecules, one different bond here, right here, compared to here, there's no extra bond. So CBD is freely rotating, where THC is locked into that conformation. That simple change makes THC interact with both the CB1 and CB2 receptors, whereas CBD does not interact with the CB1 receptor. So why? There's not a why answer there. That's just kind of the structural key component of it. And so THC, you get medicinal effects and recreational effects. Now my timing's off. But with so if you took 100 mgs of THC and 100 mgs of CBD, with THC, you're going to be in a very different state than you are with CBD, right? Like 100 mgs of THC is going to put you on the couch for 8 hours. And then 100 mgs of CBD, you could be at work, walking around, completely fine. And it's that exact same amount, the exact same amount of molecule has that big of a difference. So just kind of hammering that home here. Okay, so all of my images from papers are not showing up in this, so that's okay. We'll get to, so with the entourage effect, I'll explain it in general, and we can do it without the visuals, I would say. If you have a mixture, a hemp extract that has CBD in it, THC, CBG, there's terpenes, there's fatty acids, there's flavonoids, there's hundreds of different molecules in that mixture. So that has a different effect on your body than just one pure CBD isolate by itself. And I think in the industry, it's hyped up as this kind of magical thing that the hemp extract is uniquely medicinally beneficial. But it's very common if you start taking a step back and think about, orange juice. Drinking a glass of orange juice with sugar and water and vitamin C and all of the hundreds of different molecules is different than popping a pill of vitamin C, right? So the same active ingredient in orange juice, vitamin C, and in vitamin C tablets is the same, but the actual effect on your body and how it's taken in and the pros and cons of either type of consumption is different. And so I think it's very common to see that one molecule by itself is different compared to hundreds of different molecules mixed in a mixture. And you see that with alcohol and drugs as well. Like most drugs that interact with your brain say don't consume this while drinking alcohol. And why is that? Because now you have two different things that are both psychologically active working in the same system, and I showed you the complexity of it. So that's really what the entourage effect is. And I'm not here saying that the entourage effect doesn't exist. There's certainly a different effect when taking X hemp extract versus pure CBD. But understanding that becomes really, really complicated if you want to understand exactly why the extract is different, because there's 100 or more different molecules in there, and if every extract you get from the field, you grow hemp in Oregon, you grow hemp in Colorado, you then extract the hemp, those are different strains, they have different levels of cannabinoids, so those two extracts are not going to behave the same, even though they're both hemp extract. So from a scientific standpoint, we very much believe that there needs to be some sort of standardized study of ratios of cannabinoids and terpenes, to start to understand the mixtures of cannabinoids and individual cannabinoids themselves, versus extract A versus extract B. You're not going to get the same effect. And then from a consumer standpoint, how do you know that if you keep going to the store and buying a hemp extract off the shelf, if day to day, week to week, year over year, you're getting the same product? Because every time there could be a very different mixture of molecules in there based on where the hemp was grown, when it was extracted, how it was extracted, things like that. So moving forward, what I think the solution for that is to understand the entourage effect and to understand how multiple cannabinoids interact with each other biologically, is to do the scientific method, right? Study pure CBD, and that's actually been going on for 10, 15 years now, and we're getting a pretty good understanding of what just CBD does to the human body. But as soon as you add CBG or THC to the mix, you could have very different effects. But to truly understand that, you have to have a controlled, 50% CBD plus 10% CBG. What does that specific mixture do? And then now looking closer to, say, a crude extract, what does 10% CBG, 10% THC, and 50% CBD do? So that, you have to study that mixture by itself scientifically to understand and make real claims about that. You can't just go around saying every extract is going to have this or that effect, because it's far too complicated for that. And truly, given the amount of cannabinoids and terpenes and possibilities, there are infinite combinations and infinite effects you could get. So if you really think you have a special sauce of an extract and there's a specific ratio, great, you should study it scientifically. But the probably more useful and timely approach would be to go cannabinoid by cannabinoid. What does CBG do by itself, just by itself? What does THC do by itself? What does CBC do by itself? And once you know what they do by themselves, then you can mix the two and see what's different, right? So you change one variable at a time so you can make a conclusion, and that is the scientific method, so nothing new here. But I just think, again, there is an entourage effect that exists. A mixture of things has a different effect, but understanding it is going to be quite a effort to really understand what's going on. So with that, going to just kind of bring it back to where I think the future of this field of science is in understanding all these cannabinoids in the endocannabinoid system. Like I said, there's 100 naturally occurring cannabinoids, and they're all different keys going into this system, and each one could have a very different effect, could have a very similar effect, two of them put together. And so really it's very exciting because none of that has been done to date, and there's 50 years of advancement in science. Like what we were doing in the 1960s and 70s in science compared to where we are now in science is drastically different, and it kind of just skipped all of these very medicinally beneficial molecules along the way. So looking forward, technology has advanced 50, 60 years. It's very exciting to think how much we can learn about these cannabinoids and the endocannabinoid system moving forward, and I think there's tons of research to be done. So thank you for listening to me spiel about science all morning, and take some questions.
All right.
Oh, he's got the mic.
Two questions.
Mm-hmm.
What's the difference between a phytocannabinoid and a cannabinoid?
Yep.
How should a marketer talk about that?
Yep.
And second question, CB1, CB2 receptors, THC and CBD interact with one of them, but they both do not interact with the other, correct? And remind me of what that was.
Yeah, so two questions there. kind of getting back to the terminology, and that's where my picture wasn't there, so I didn't touch on it as much. Cannabinoid refers to all cannabinoids, so endocannabinoid or phytocannabinoid. So it's the term dog, right? That is a dog. Now, phytocannabinoid refers to, say, Cocker Spaniel, and endocannabinoid, different breed of dog. So endocannabinoids, endo meaning it occurs in the organism, so it occurs in the human body. Those are the endocannabinoids. Phyto, I'm probably Greek there, phyto relates to plants, so phytocannabinoids come from plants. They're all cannabinoids under the term. So that's the strict jargon there. And then receptor-wise, let's go to my first slide with two things on it. So yeah, that's not the right one. Yeah, this one. So THC interacts with the CB1 and CB2. And CB1's the one in your brain, memory, cognitive function, things like that. So that is why THC gets you high. Both of them, CBD and THC, interact with the CB2 receptor, which is in your peripheral nervous system and inflammation response system. So, yep. Whoever. Yeah, just go around. I think we've got 10 or so minutes. So.
Hi. If prior to hemp coming into mainstream, how did the human body satisfy the needs for these keys?
Yep.
Were there other foods or?
So the current medicinal understanding is, like I said, you have endocannabinoids in your body, and I didn't show them. Again, that was the picture that wasn't there. The primary ones are called anandamide and some weird chemical name. It's 2-AG for short. So anandamide, AD, and 2-AG are your body makes those chemicals and uses them to regulate the endocannabinoid system. So like when you get a runner's high or you get poked with something, that whole biochemical thing is happening naturally. You have a system to do that, to make anandamide, send it out, stimulate the receptor, treat the pain, inflammation, and then kind of go back to stop making it. So your body quickly makes it and then breaks it down in a direct response to some sort of stimulus. So when you add the phytocannabinoids, THC or CBD, to your body, you're essentially artificially like stimulating that receptor without the biological, say, response. But that can help because you have inflammation and it's getting worse and your body's not keeping up with it. So by adding, say, CBD there, you're telling your body to, say, deal with that inflammation more. And that's where the medicinal effect of it comes. So it's an extra additive that, say, stimulates more of the body to do that thing.
Thank you.
Yes, I had a question about the future of other cannabinoids. I know CBG is essentially the next big cannabinoid that's being researched more heavily. After that, out of these other dozen or 100 cannabinoids, which one would you say is being the most focused on as a priority for research that would be kind of following up CBG as maybe the next big extractable ticket?
Yeah, I actually think it's very driven by how the plant works. So the plant works by making CBG, turning it into THC, CBD, or CBC, and then THC or CBD eventually end up being CBN. So that's kind of how the plant makes them. So these 5 are the big ones, I think, of interest because of their availability. The plant makes these 5 the most. All of the other crazy ones are really, we don't think, synthesized like by the plant. Something else like happens chemically. So like adding oxygen, that's the oxidation. That's a very common chemical process, but that's what a lot of these are. And then all of these random structures are just kind of spontaneous bond formations happening. And so they're much rarer because the plant's not like synthesizing them, I would say, quote unquote intentionally. No one knows the intention of a plant. But anyways, those 5 that I showed, CBG, CBC, CBN, would be the other 3. And I think it's because they occur the most and they're the most accessible, whereas all the other ones are just harder to get to. And that's kind of from an R&D standpoint what we're very focused on. But any one of them, you can't predict which one is going to have the effect. We've been trying for 50 so years to predict that using computers to model. You can model all of these as molecules in that simulation, but you get into like quantum mechanics and computers can't do that, and you can't actually predict it a priori very well. So who knows? Yeah.
Hi. In terms of the other cannabinoids, besides THC and CBD, are they all interacting with the CB1 and CB2 receptors, or are they doing other things?
Yeah, they probably are, and there is evidence that CBG, CBC, CBN work on those receptors, and just based on the structure, and they're similar keys, right? They're not vastly different keys, so it's a good guess that they're interacting with those receptors. So that's the current model, right? And I think that's a reasonable guess. There's also evidence of other actual cannabinoid receptors that have yet to be characterized. So there's probably a CB3 receptor, and again, just the science has a very difficult time like characterizing and locating those receptors, but there's evidence that there's other receptors that uniquely interact with these as well.
And I was also just wondering about developing tolerance and how the mechanism there, and if there's any strategies around that.
Yeah, I think that's just the kind of standard biochemical response to most things. So like the question a couple minutes ago, you're say lacking in something, and this works for alcohol or opiates or cocaine, whatever drug you pick, you do it, you get an effect, so then your body changes how it's producing chemicals to counteract the effect, so then you do more, or so it's very complicated, and I think there's tolerance to some degree with THC and CBD and all cannabinoids.
My question is, does the endocannabinoid system only accepts cannabis molecules or other vitamins as well?
Yeah, it accepts other molecules as well. So terpenes are known to interact with it, and those come from plants all over the animal kingdom.
And so the endocannabinoid system was named after the plant?
It was. So the endocannabinoid system was named after cannabis because they found that those molecules interacted so strongly with it. So that's how the name came. They knew about cannabis interacting with it before they knew what the receptors were. So that's where the name came from. But yeah, if we can pass it across to this side. Sorry, everyone over here. We've got 4 more minutes-ish.
Could you elaborate on where you said that CBD is primarily interacting with the CB2? So I'm assuming that it's still reacting with this, yeah?
Yeah, so it gets very this is now where you start getting towards, where is this picture? I think I'm going the wrong way. This sort of picture.
So is it wrong to say that CBD works on the CB1 receptor? Because it
is pretty much, I would say, like, wrong. And so the way you fundamentally scientifically think about it is like you get this number, it's called your binding constant, and it's a measure of how much the key is stuck in there. So if a key gets really stuck in there and you can't yank it out, that would be a very strong binding constant. And then if it goes in and comes right back out, it's a very weak binding constant, and that's like your main variable for any molecule binding to anything. So you could look at how vitamin C binds to CB1 and get that number, but it would be a very low binding constant. And so it's kind of a relative sense of what a big number means a lot more for binding constants, like very tightly bound. Yeah.
Hey, thanks for the talk. 2 things. Is it possible to get a copy of your slides?
Yeah, probably.
Second, so I understand that you can chemically remove THC. And then to your knowledge or to the extent of your experiment, are you able to remove just THC without touching any other bonds? And
yeah, so using different chemical processing, you can have an extract that has however many compounds, and you can isolate individual molecules within that mixture. And that's been the practice of natural product chemistry for going back hundreds of years, where like alchemists way back in the day would have a mixture of plants and do some different chemical things and get just that molecule. And that's how, again, I keep using cocaine as the reference, but that's how cocaine's made out of the coca plant. They do all these extraction steps. Same with caffeine coming from plants and going into your soda. Take the caffeine from the plant and isolate it. So yeah, it's possible. And
Okay, so we have time for one more
one
question.
more. All right.
So is there a difference in the bioavailability of topical CBD versus edible CBD?
Yeah, so bioavailability gets, again, into, you keep going down this rabbit hole of complexity, but where and how the molecule is delivered does matter. So if you put it on your skin, your skin tries to block things out, but some things go through it better than others. So that's one aspect. If you eat it and it goes into your intestines or your stomach first, it's exposed to a very acidic environment. That can start breaking things down and changing molecules and stuff as well. If you inhale it into your lungs and it goes straight into your blood system, if you put it in your lips like tobacco or something. So all of those modes of consumption do change the bioavailability of it, and you can add things to the mixture to change the bioavailability as well. So, cool. Well, sounds like we're out of time, but thanks everyone for coming.
Thank you so much, Jake.